This is the revision video for Unit B3, Growth and Development. All living things are made of one or more cells. Cells are very small and can only be seen with a light microscope. Plant and animal cells have some features in common, but plant cells have some features that animal cells don't have. Both plant and animal cells have a cell membrane, nucleus, cytoplasm and mitochondria. Cell membrane is a thin layer around the cell that controls the movement of substances in and out of the cell. The nucleus is a structure inside the cell that contains chromosomes made of DNA. The nucleus controls the activities of the cell and how it develops. Cytoplasm is a jelly-like substance that fills the cell. Most of the chemical reactions of the cell occur here. Mitochondria are small rod-shaped structures that release energy during aerobic respiration. Plant cells also contain a cell wall, permanent vacuole and chloroplasts. The cell wall is a layer on the outside of the cell membrane. It is made of cellulose which is strong and supports the cell. A permanent vacuole is a fluid-filled cavity containing a liquid called cell sap. Chloroplasts are small disc-shaped structures found in the cytoplasm of some plant cells. They contain a green pigment called chlorophyll, which traps light energy for photosynthesis. Chloroplasts are found in cells of the leaves and stems where photosynthesis can occur, but are not found in the cells of the roots or the flowers. Bacteria consist of one cell. They are smaller than plant or animal cells and have no nucleus, no mitochondria and no chloroplasts. Despite having no nucleus, they do still contain DNA in the form of a single strand or chromosome floating in the cytoplasm. So how do these organisms grow? Plants and animals start as one cell. This cell is an undifferentiated stem cell. It divides by mitosis to give cells which can then become specialized. The process of cells becoming specialized is called differentiation. Stem cells can develop into any of the different cell types and form tissues and organs. Because bacteria are only single cells, they can grow and divide very quickly. They divide into two, and then each new cell increases to full size. Division may happen every 30 minutes if there is enough food, oxygen, space and a suitable temperature. In this way, the population of bacteria grows very quickly. Animals only grow in the early stages of their life. They will grow until they reach full size and then stop growing. In humans, there are four main stages of growth. During infancy, growth is rapid following birth. During childhood, growth slows to a steady rate. During adolescence, puberty causes rapid growth. Finally, during adulthood, the growth rate falls to zero. Plants grow continually throughout their lives, but they only grow at specific points: the root tips, the shoot tips, the stem nodes and the buds. These parts contain meristem tissue, which contains stem cells. Grass also has meristem tissue in the leaf blades, which is what allows it to grow again after grazing or mowing. Differentiated plant cells cannot divide due to their thick rigid cell wall, so plant growth occurs at particular parts where meristem tissue is found. Meristem tissue contains stem cells which are undifferentiated, have very thin walls, are small and do not contain chloroplasts, have very small vacuoles, and are packed closely together. So, in summary, plants and animals start life as one undifferentiated stem cell. This cell divides by mitosis to give cells which can then become specialized. This process of becoming specialized is called differentiation. Stem cells can develop into any of the different cell types and form tissues and organs. Human differentiated cells can divide to repair tissue damage, but cannot differentiate into any other type of human cell. Plant differentiated cells cannot divide because they have thick, rigid cell walls. Medical research is developing ways of using stem cells. Medical researchers can get stem cells from spare very early embryos created during IVF treatment. They can then make them differentiate into any type of cell by giving them an appropriate chemical signal. They are developing ways of using these cells to treat people with Parkinson's disease, repair spinal cord injuries so people can walk again, grow tissues or organs for transplanting, and treat people with type 1 diabetes. Scientists can also obtain stem cells from bone marrow or umbilical cord blood, but these stem cells cannot differentiate into as many different types of cell as embryonic stem cells. However, the use of embryonic stem cells raises some ethical issues.